Hi, my name is Shoshana Daly, and my project is titled The Rational Design of a Lipase from Geobacillus Stereothermophilus T6 for Increased Stability in Methanol. I know, that title's a lot. I'm going to break it down. So basically what that means is I was testing the stability of a mutant enzyme to see if it was more stable than its wild-type counterpart when in methanol. And this work was done in the hopes of developing an enzyme or process that could facilitate a more efficient biodiesel production. So what does methanol enzymes, what does any of it have to do with each other? It's all tied together with a process known as transesterification. So transesterification reaction is a reaction used to produce biofuel, biodiesel more specifically. In the reaction, a triglyceride, in our case, waste chicken oil, so this triglyceride reacts with an alcohol and is catalyzed by the lipase, a type of enzyme, to create fatty esters and glycerol. One more time, that was complicated. Triglyceride reacts with alcohol with the help of lipase to create fatty esters and glycerol. So these fatty esters, also in this case known as fatty acid methyl esters because we're working with methanol, is what can be refined into biodiesel. So why did I do this project? What does transesterification have to do with anything? So I completed this project as a part of the 3-year science research course at my school, but it was more than that. I went into the course really passionate about climate change. I'd been speaking and doing activism for a few years at that point, and I still do those things. And so I went in and I was like, I'm going to focus on carbon capture. But the reason I wanted to look at carbon capture is I was like, seems like magic. We can just take the carbon out of the air, solve the whole problem. As I looked more into it, I saw like this isn't a magical solution. Carbon capture is just a way to defer from the emissions that we keep putting out, and the real solution is to reduce our emissions and turn to sustainable energies. So I started researching sustainable energies. I looked renewable energy, and I looked at solar and wind, but I eventually biodiesel caught my attention. I'm also fascinated with biochemistry and biochemical pathways. I think that the way that our bodies and chemicals and all these outside parts come together to let us and animals and plants function is just absolutely amazing. And so my fascination with biochemistry and biochemical pathways came together with my interest in and research of biofuels, and I looked at enzymatic biodiesel production. So, what's the significance of my project? Biodiesel is a bridge to sustainability. The big difference between biodiesel and other sustainable technologies like solar and nuclear and wind is that solar, nuclear, and wind, they need an energy storage mechanism. We need new infrastructure to be able to capture the energy that they're producing and efficiently get it to the people that need it. And while this is a possibility in the future, the technology needed to harness and project this energy is not where it needs to be yet. And the thing with biodiesel is that it's compatible with current technology. We don't have to change anything. We just have to make the biodiesel cost competitive with crude petroleum. It works in the combustion engine that's already in trains, boats, barges, etc., etc. So how has this project helped me to develop my talents? First, I had to read a lot of scientific journal articles at the beginning of the project as I tried to hone exactly what I wanted to research and who I wanted to work with. And through this, I developed my skills to read these journal articles because it was hard for me at first. I tried to understand every little word. Obviously, that's not how you want to go about reading a journal article. What I learned was to skim the papers and then break down sections that would be very pertinent to what I wanted to research, and then break down vocab and read it again, annotating along the way. Also, I learned great communication skills between the presentations I've had to put together, and also communicating with professionals. So I had to reach out to a bunch of professors and grad students when I was trying to secure a mentor, and learning to write these types of professional emails and communications was very helpful. I feel like I really learned how to do this type of thing, and it's very useful when I will go and eventually work at university or grad school or just in the future. I also learned more specifically lab work and techniques when I was in the lab. Firstly, I learned purification of enzymes. I broke cells using an emulsifier to separate the enzyme. I used the Bradford technique to check for the purification in early stages. I used an active device to separate the protein from that specific protein from everything else in the cell. And then I used SDS page to again check the purification of my samples. I also learned to work with a centrifuge, and I learned to conduct activity assays and use a multiplate reader and use a hood and other different things. So I also learned scientific writing. When I went into this, I really was not confident in the field of scientific writing. But between writing my research paper and specifically developing my review of literature and procedure, I felt like I really progressed and I really developed skills that will be useful for whatever research I end up doing in the future. And finally, might be kind of cliche, but I feel like I really learned perseverance. Um, I really didn't realize how much failure was involved in the field of science, and just so I wrote to a ton of mentors. Took me many communications before I got to one that would let me into their was nice enough to let me work in their lab and provide me with someone to supervise me. I also had many troubles and had to troubleshoot a lot of things when I was in the lab, and just learning to persevere when it comes to writing, doing applications, writing papers. I feel like that was a skill that I very much gained from this project. So, how does my submission fit into the larger context of my field of study? So, I feel like, kind of said before, with the uh, biofuel is a bridge to a sustainable future. We can use it to fight climate change if we can just get it to be industrially viable, which just we need to make it efficient, the production to be efficient. But separately, it's part of the field of biocatalysis. How can we manipulate enzymes to change biochemical pathways to produce things that we can use as humans or that plants can use? We can change these pathways by changing the enzymes, and this is part of that. I used rational design, which is using computers to predict where the mutation should be. And really, I think a big question for the future of this field is what's what's the most effective way to manipulate these enzymes and when. So finally, or not so finally, semi-finally, I did my work in the Fishman lab at the Technion Israel Institute of Technology. And the work that led me to do my project was done by Dr. Fishman, who ran the lab, Shalev Jihaz, Inbal Rush, Yael Bash, among other scientists. They developed earlier versions of the lipase I was working with, and then I added on to their research. I really hope to become a medical researcher or a doctor. I really want to help people. Something I've always dreamed of doing was participating in Doctors Without Borders. And yeah, that's my project. I really had a great time working on it, and I hope you will consider me for the Davidson Scholarship. It would be an honor.